Hi, everyone. It's really a pleasure to be here to talk to you about some work we've been doing using sequence-based approaches for natural product discovery. So I think I probably don't need to tell you that natural products have been an important source of medicine throughout modern history. Most of these molecules have come from plants or microbes derived from soil. Starting in the 1970s, there was a major effort to look at marine natural products as a source of potential pharmaceuticals. And this largely involved collecting various types of soft-bodied animals, animals that use these molecules as chemical defenses. And these efforts were actually quite successful. To date, there are at least 14 marine natural products that are approved as drugs. A few of those are shown here. Many of these are used to treat cancer. These molecules are often cytotoxic. They're used as chemical defenses against predation by the soft-bodied invertebrates. And again, a couple examples with ascidians and sponges. This is a shell-less mollusk that's lost its shell because it produces a chemical defense to deter predation. An exception is the cone snail, which produces a series of peptide toxins that are used as venoms for capturing prey. And so, despite these successes, as you might imagine, it can be particularly challenging to develop a marine natural product as a pharmaceutical due to supply issues. Many of these compounds are structurally complex, difficult to synthesize in any sort of commercially viable way. And that's hampered the field in many cases. And so how do we get around that? Well, one way is to target marine microbes. And if we can culture them, then obviously they become a renewable resource for natural product discovery and research. And so we've been particularly interested in microbes that live in ocean sediments. Sediments cover the majority of our planet's surface. There are vast areas of deep ocean sediments that have yet to be explored in terms of microbial diversity and natural product potential. We know these communities are diverse in terms of their communities. And so we've been really keen about the idea of studying these microbes and their potential to produce natural products. And so we've done that over the years in a variety of ways. It might simply mean collecting samples by hand. We have used commercial fishing gear and small sediment grabs to collect samples even as deep as 1,000 meters. And we've used oceanographic research vessels to collect samples even deeper. And also some remotely operated vehicles lately we've been using to get some really deep samples. And so we can then use those samples and selectively cultivate bacteria that we're targeting. And in our case, we've been particularly interested in actinomycetes. And as you might know, this group of bacteria is common in soils and known as a good source of antibiotics and other natural products. And so one of the first questions we wanted to ask is, if we go to the ocean and we look for actinomycetes, are they different from those that we see on land? And in fact, we were able to see that there are quite a few diverse groups of actinomycetes that do look quite different from those on land. I'm just going to tell you a few words about one of those lineages. This is a genus that we've described as Selenospora as the first obligate marine actinomycete genus, because this group failed to grow when we removed seawater from the growth media. So they have some clear adaptations to life in the ocean. To date, we've named 9 species, and we've been studying these bacteria extensively as a source of natural products. And so to date, this is the collection of molecules that have been found from this genus. I want to give an acknowledgement to my colleague, William Fennicle, an organic chemist who I've worked with for many years on natural product discovery. He led the effort to solve the structures of most of these molecules, although other groups have worked on this genus as well. And so to date, more than half of the molecules that have been found from this genus are new, I think, which would support the idea that if we can find new taxa in poorly explored environments, then that's a good strategy for natural product discovery. And I'll just highlight one of these molecules quickly, Sinosporamide A, because it is currently in phase 3 clinical trials as an anti-cancer agent. It's a potent proteasome inhibitor. The original indication was targeting multiple myeloma, but we now know it crosses the blood-brain barrier, and it's being tested against glioblastoma. And we're hoping that we're going to hear some good news about these phase 3 trials by the end of this year, in which case this would be the first marine microbial natural product to be approved as a drug. So that's pretty exciting news that we're following, and hopefully you'll hear more about this later. So all of these efforts were made using what I'll call a traditional discovery paradigm, culturing strains of bacteria, growing them in the lab, using organic solvents to extract the natural products they make, test those extracts in some sort of bioassay, use that bioassay to guide the isolation of active molecules, solve the structure of those molecules, and hope they're new. And so that can work. It's sort of a tried and true method, except one of the challenges is that now we constantly find common bioactive molecules, and these become nuisance compounds, and it becomes more difficult to find the real gems amongst all of this background. And so, of course, today in the genomic area, we can take a different approach. And the field has really been transformed by the ability to sequence genomes and take those genome sequences and run them through existing bioinformatic pipelines, such as anti-SMASH, where the genes that encode the enzymes that assemble these natural products can be identified, and they can be compared to known experimentally characterized biosynthetic gene clusters using programs like MyBig. They can be put into a sequence similarity network using programs like Bigscape, and put into a phylogenetic context using Corazon. And so there's a lot of opportunity to use this type of bioinformatic approach to assess bioinformatic potential prior to doing any chemical analysis. And then based on this information, we might determine, oh, it looks like a strain is able to produce a compound that's known and we're not interested in, so we're going to avoid that strain. Conversely, a strain may have some interesting biosynthetic potential, and we then may focus on that strain either by culturing and trying to get the gene cluster of interest expressed, or using heterologous expression or some other type of genetic engineering to get that molecule expressed and identified. And so we got into this game quite a long time ago. We sequenced one Selenospora genome back in 2007. And so you'll see Ala Lapidus is a co-author on this paper. She's one of the co-organizers of this conference. And while she was at the Joint Genome Institute, she helped us assemble and annotate this genome. And at the time, we were amazed to see that about 10% of it is devoted to specialized metabolism. And at that time, these bioinformatic tools that I just mentioned, like anti-SMASH, didn't exist. So you had to manually go in and try and find these gene clusters and annotate them, which we did for this genome. And we identified a gene cluster where we didn't know the product, and we're able to interpret it and make some predictions about what the structure might look like. And this is what these tools now do automatically. But in this case, you can see the complexity of, in this case, the polyketide synthase genes that encode the biosynthesis of this molecule. You can see that they have this highly repetitive nature, which we won't go into details in this case, but we can read this and make predictions about structure based on this gene cluster. And in fact, we were able to help solve the structure based on the predictions of this gene cluster. And interestingly, because as you might imagine, you can see there's a lot of repetitive elements here. This can create challenges with assembly. Once we had a confident structure, we could go back and look at the gene cluster and see that some regions look like they might have been collapsed, and we're able to resolve those. And so that was an early effort, and genome mining now has become enormously popular, a standard tool, I would say, in the field of natural product discovery. It's extended beyond genomes, though, to metagenomes. There's been a couple of recent high-profile papers where MAGs, or metagenome assembled genomes, have been studied from environmental samples, and it's been shown that there are various taxa that are enriched in these types of biosynthetic gene clusters that haven't really been studied in the past. And so one example is acidobacteria, where you can see they were able to show that they had over 150 different biosynthetic gene clusters in one of these acidobacteria MAGs. And so pretty extraordinary amount of diversity. Sorry, that's a total of over 150. These can contain up to 15 different BGCs in an individual MAG. And so really exciting opportunities here, but as you might imagine, we're only assembling MAGs from the most abundant genomes. And so we're gonna miss some of the rare biosphere, which can be important in terms of natural product discovery using this type of approach. Just one other study I'll mention, this from the JGI as well, and a recent really expansive study looking at over 50,000 MAGs from over 10,000 metagenomes. And they were able to identify huge numbers of biosynthetic gene clusters using these automated approaches. But what I want to point out is that many of these gene clusters are on contig edges. And you can see that here they're showing that large numbers of these are fragmented. And when they're fragmented, they become a lot less useful when analyzed by tools such as anti-smash, which are really designed to look at the entire gene cluster. And so that leads to another concept where instead of targeting the entire gene cluster, what if we just target specific sequence tags to try to get some sense of biosynthetic potential? And so in this case, the sequence tags we're talking about are associated with polyketide synthase and non-ribosomal peptide synthetase genes. These are involved with making lots of important natural products. The sequence tags specifically are the ketosynthase domain in the PKS gene and the condensation domain in the NRPS gene. And these are really the action parts of these genes. The KS domains are covalently joining the extender units in polyketide biosynthesis, and the condensation domains are catalyzing the peptide bond formation in non-ribosomal peptide biosynthesis. So we've known for quite a long time that we can put these sequence tags into a phylogenetic context and really learn a lot from that. So here's a phylogeny from this paper from Michelle Moffat almost 20 years ago now. And what you can see is that there are very distinct clades. We can distinguish type 1 polyketide synthases from those that are hybrids shown here. And we can see within type 1 PKSs, we can recognize various groups of microbes can be distinguished as well. And so we got really interested in this idea of using sequence tags and phylogeny to make predictions about natural product biosynthetic potential. And almost 10 years ago now, we released a tool that we call the Natural Product Domain Seeker. And what this tool does is it assesses PKS and NRPS diversity based on these KS and C domain sequence tags. And so the concept behind this tool is we developed a database of KS and C domains from experimentally characterized gene clusters. We put those into a phylogenetic context, and that's shown here. And then we looked at the clading patterns and we asked how the clades corresponded to the known functions of those KSs and the architecture of the gene clusters that they're in. And so we can clearly see delineated type 1 PKSs, type 2 up here. We see those involved in fatty acid biosynthesis. And within type 1, we see a variety of subclasses, trans AT PKSs. The AT domain is not actually in the module. We see KSs involved with making things like enedianes up here. We'll talk more about those in a minute. So we're pretty excited about being able to basically classify gene clusters just based on these sequence tags. And so what the tool does, you submit your query data. It can be nucleic acid or amino acid. These could be PCR products or genomes at the time, is what we were thinking about. And basically, the tool will blast your query data against the database. It will identify KS or C domains in your query data. It will tell you the top blast match to the internal database, give you an E-value and a sequence similarity to that top match. And you can output trimmed sequences. You can output alignments, and you can put those into a phylogenetic context as well. And so just to put this into a little context, here's the biosynthetic gene cluster involved with the biosynthesis of erythromycin. Okay, so these are very large gene clusters. There are lots of genes that do things like tailoring, maybe adding a sugar, that are involved with resistance or export, transport, all kinds of things. In this case, there are 3 core biosynthetic genes, 3 polyketide synthase genes that really do the building of the core molecule. And so within each of these PKS genes, which are shown here, there are modules which are in these boxes, and the modules have various domains in them. So we're just targeting the KS domains, and there are 7 of them. All we'd have to do is detect one of these 7 KS domains using this tool. And if by comparing it to the database, if there was a close match to the erythromycin KS in the database, then you would be able to make a prediction that this gene cluster is present in your sequence data just based on this sequence tag, okay? And so a pretty quick and facile way to make a prediction about pretty complex sequence data. Okay, so we're hopefully soon going to release version 2 of this web tool. We're beta testing it now. What we've done is added considerably more ketosynthase diversity. There's lots of new functionality that's been discovered since the original release. We've increased the taxonomic coverage, so you can see we've added almost 4 times the number of sequences. We're only updating the KS workflow right now. And so we've been able to, based on the new phylogeny, increase the number of KS classes and subclasses we can identify in that phylogeny. And so this is basically what it looks like. And so once again, we can see these major clades, type 1 PKS, type 2 PKS, FASs. We're now able to distinguish FASs of bacterial and fungal origin, or metazoan and protist. We've dramatically expanded how we're able to classify type 2 PKSs by dividing them into quite a few different classes and subclasses, which can be seen here. So this adds a lot of information to the output. And again, with the type 1s, we've further expanded the classes that we can recognize, including recognizing those of fungal origin and those involved in making certain unusual classes of natural products that to date are quite rare, such as, again, this enediene category. And so we've increased the file size to 500 megabytes and 500,000 sequences to better accommodate amplicons. We've replaced BLAST with Diamond, which has improved the speed with which the program works. And we've created some new functionality that I'll take you through, and including what we've called a result summary page and a BGC ID card. Okay, so let's run an analysis. This is what the website looks like. You're going to have to select if you want to search for KS or C domains. So we select KS domains. If it's amino acid or nucleotide sequence data, and so you have to select that. You upload your sequence data, hit seek, and what you're going to get initially is a summary page. And we added this because now with larger data sets, you're getting so much data, it's difficult to just scroll through it all, or it may not get you where you want to go. So now we can see, so here we've uploaded one genome sequence. This was a closed selenosporic genome sequence, so there's just one sequence. 28 KS domains were identified. And now here we can see the breakdown of the classes and subclasses that were detected in that genome sequence, okay? And so this is pretty useful. You may be interested in a certain group here. And so in this case, we're interested in enedienes, okay? So we can select that and we can view that subset of the data. And this is what we see. And so there were 2 of these enediene KSs detected. We can see the query ID here. Appended to it is some information that tells you about the position of that domain within your query data. There's information explaining that on the website. It gives you the top match in the database. So that's to a KS and sporolide. The percent identity is 100. Okay, it gives you the alignment length, the E-value, the product of that gene cluster sporolide, and then the class and subclass delineation. So we know that this strain actually produces sporolide, so it doesn't surprise me that we get 100% match of the KS. Then we see a second domain with a much lower class identity for the KS at 65%. The best match is dynamycin, so we would predict that this strain is making a second enediene, probably not dynamycin, probably something that may be new and hasn't been experimentally characterized yet. And so we can do a number of things from here. I mentioned this BGC card. We can click on sporolide, and that takes us to a lot more information about this molecule and its gene cluster, where it was originally found. There's links to the PubMed ID, also to the MyBig ID, which has the complete gene cluster. We also list all of the KS and C domains that are in that gene cluster and their class and subclass. Okay, so there's only one in this case, and it's an iterative type 1 cis AT KS. So if we click on the MyBig ID, that can take us through MyBig to the anti-smash results. And so now we can see the full gene cluster. So again, a very large gene cluster. It contains one polyketide synthase gene shown here. And now we see a breakdown of that polyketide synthase gene. So there's the one KS domain that we detected, right? And so this is our sequence tag that's giving us pretty good information that this gene cluster is probably going to be involved in making an enediene. And if the match is close to something in the database, there's a good chance it's going to be making molecules in that specific compound class, just based on that single sequence tag. And so you can also go to the BGC tab, and you can scroll through all the various gene clusters that are currently in the database, and you can download all of those KSs as well if you want to generate your own phylogenies, which we highly recommend. Okay, so lots of information from these very short sequence tags. And the question then becomes, how well is this really working, right? And so for this second release, let me just acknowledge Lisa Clow. I have her name there because she did a lot of this work assembling this database and helping to put version 2 together. Okay, so how well is this really working? So we spent a lot of time testing the detection accuracy, and I'll acknowledge Caitlin Creamer here, a PhD student in my lab who has been leading this effort. And so what Caitlin did was assemble a lot of positive controls. And so those are the KS sequences currently in our database that we know what they do. And then she went to the larger condensing enzyme superfamily to which these KSs belong and selected a large number of negative controls. And then the question was, how often does NAPTOS detect false positives at various e-value cutoffs? And so here's a plot showing that. So the first thing we see is that NAPTOS2 is considerably detects considerably fewer false positives than NAPTOS1 at an e-value of -10. NAPTOS2 doesn't detect any false positives at an e-value of -5, detects 14. And so what we decided to do was select as the default setting e to the -8. It maximizes sensitivity. All of the positives were, all of the true positives were detected, and this minimized false positives to 2.8%, which we viewed as being an acceptable rate. So the next question is, there's detection and there's also classification accuracy, and that's going to be affected by sequence length. And so we tested that as well. I'll acknowledge Sheila Podell. She's a programming engineer at Scripps who's been instrumental in building this web tool and helping us test this second version. And so here, Sheila's tested different lengths of amino acids and in KSs. And so KSs are generally around 1,200 nucleotides in length, 400 to 450 amino acids. And so in many cases, you may not get even the whole KS, right? So what Sheila did was she tested overlapping subsets of these different amino acid lengths, covering the full KS sequences. And so this would be to sort of mimic what you might see in fragments in a fragmented genome or in a metagenome. And so what you can see is that at a length of 200 amino acids, okay, so a little less than half of a full-length KS sequence, we're still able to accurately detect about 99% of the true positives, and we're able to accurately classify about 85% of them. So we're pretty happy with that, and you can see that these values fall off pretty dramatically as the sequence length gets smaller. And so based on this, we set the default minimum amino acid alignment length at 200. Okay, so that's basically the tool and how we establish the default settings for it. So you have a sense of what it can do. It can identify these sequence tags and various types of sequence data. And so now I just want to show you a couple of examples of the application of this tool and why we think it's of value. And so the first is for genome sequences. And so we've sequenced to date 118 selenosporic genomes. When we run those through NAPTOS1, all we can do is detect how many type 2 PKSs are present, okay? And so these are the numbers for the different species. With NAPTOS version 2, first we can see that we detect many more type 2 KSs, so the tool seems to be working quite a bit better. And we also are able to classify them into these various groups. So we get a lot more information, more specificity about what types of molecules these type 2s may be producing. And so that can be particularly useful if you're looking for certain groups of compounds or certain types of molecules, or if you want to compare different species and see which ones may be more enriched in this sort of biosynthetic potential. So lots of applications for mining genomes. Okay, it's also really well designed for amplicons. And so here we've taken a data set, an existing data set from a paper that was recently published in Microbiome. And here they've used PCR primers that are targeting type 2 KSs. And so, you know, as we know, PCR primers aren't always 100% specific in their targets. And so why this tool is particularly useful is we can really get a sense of how well those primers have worked. And so in this case, they looked at soils from a variety of locations. We're just showing 4 here. And they generated large numbers of PCR products from those different soils. And their interest was really to compare, do sort of a biogeographical comparison of these different soils, and that's fine. But you can see when we ran these amplicon data sets through NAPTOS2, in many cases, many of these amplicons weren't identified as type 2 KSs using NAPTOS. And so that could mean that they're really new and different type 2s, which would be great. And it is worth exploring because that means they may be making some really new types of molecules that we don't know very much about. Alternatively, they just may be off-target products, and so they're not actually involved in natural product biosynthesis at all. And so, and of the sequences that NAPTOS2 detected, you can see in some cases that a lot of these were FASs or involved in fatty acid biosynthesis. Okay, so that's important to know. If you're interested in FASs, that's great. If you're interested in antibiotic discovery, this isn't particularly useful for you. Okay, so we're able to I distinguish those from all the rest of the type 2s and the various subclasses that are in these different sample types, which can then be compared. So you can see how this is a really fast and straightforward method to analyze amplicon libraries. And there really aren't any existing tools right now that can do that type of analysis with amplicons. And I want to acknowledge Caitlin Creamer here, a PhD student in the lab, who's really done a lot of great work on these, what we're calling use case tests of the tool. And then the last application I want to talk about is metagenomes. And here I want to acknowledge Han Singh, a PhD student in the lab, who really got interested in trying to apply this new release of NAPTOS to existing metagenomes. And so he downloaded 240 gigabase pair of metagenomic data from the JGI website. And he was really curious because they encompass different biomes, and he wanted to know how these biomes differed in terms of their KS composition. And so he focused on type 1 KSs, and he used the default settings in NAPTOS, and he used the metagenome assemblies, because if we use the unassembled data, we end up with a lot of very short sequences, and we already know that we're not going to be able to detect and classify those very accurately. And so he took all of this data and he generated a PICO-A plot using a Bray-Curtis dissimilarity matrix. And we were really pretty amazed to see the distinction. So here the colors represent the different biomes. You see a lot of blue here, and the blue is largely soil and rhizosphere. And then we were very excited to see all this red way over here and this red is largely marine sediment. So it really looked like, in terms of type 1 chaos diversity, what was seen in these marine metagenomes, sediment metagenomes, is very different from what's seen on land. And so Hans then wanted to know what was driving those differences, and what he found was that certain groups of chaoses really seem to be distinct in these marine samples, and in particular, those encoding indienes. And so now what he's done is he's downloaded the chaoses from Naptos that he's detected in these metagenomes, and he's put them into a phylogenetic context, which is shown here. Okay, and so again, the color coding is by biome, and now in black, we have sequences from this MyBig database of experimentally characterized indiene gene clusters. And so you can see there's a lot of blue here, all the soils, everything that we really know about is coming from these soil samples. And now we see that these marine sediment chaoses are really distinct phylogenetically, and nothing in here has been experimentally characterized yet. So a lot of opportunity for finding new indiene type molecules from microbes living in ocean sediments. And so why is that important? Well, as of 2015, only 11 of these indiene molecules have been identified. 2 of them are used in the clinic as anti-cancer agents. One of them is calicheamycin, the structure of which is shown here. This is the indiene portion of the molecule, which gives it its name. This is a very reactive ring structure. It goes through something called a Bergman cyclization. It abstracts hydrogen from DNA, causing a double-stranded cleavage in DNA. And so these are some of the most potent cytotoxic molecules known. And in fact, the way they're administered is through linking them to an antibody in what's called an antibody drug conjugate. And so in this way, these highly potent cytotoxins are targeted directly to a cancer cell. And so there's a lot of interest in finding more of these molecules, and it looks like microbes in ocean sediments may be a great source for them. And so then the question is, which microbes are these chaoses housed in? And so Hans addressed that as well. And so here, once again, we have the chaos phylogeny, but he's added this time chaoses from genome sequences. So indiene chaoses from genome sequences, and that's in the inner ring. And the inner ring is colored by who the bacteria are. And so here you see these are all actinobacteria, and you see in the outer ring, these are the metagenome sequences, and these are all soils. So it's looking like, you know, the soil chaoses are likely in actinobacteria. And then when we come to the marine chaoses over here, we see that they're clading mostly within this sort of purple group, and purple are proteobacteria. So proteobacteria are not really well known for making indienes. So it looks like whoever's living in ocean sediments and potentially producing indienes, it may be proteobacteria. So it gives us a sense of possibly who to target if we're interested in trying to culture them or try to assemble genomes that contain these gene clusters. And so Hans also explored the cis-AT KSs that were in all these metagenomes. And so now we're taking all those sequences and we're putting them into a sequence similarity network shown here. And again, color-coded by biomes. And so we see lots of blue and black up here. These are the soil KSs again, and the black ones are the ones that we know what they do. These are in the MyBig database. Then we see lots of other colors scattered throughout the plot, including a lot of yellow up here. This is really interesting. These are host-associated, suggesting that there are a lot of unexplored cis-ATKSs in microbial symbionts. And so, and then Hans's question was, who are all of these in? And again, the soil cluster is largely in actinobacteria. I guess that's not surprising. But if we look at all of these other KSs, which are in these other biomes, they're in really scattered among many diverse groups of bacteria that aren't really recognized as producing natural products or polyketides. So again, lots of opportunities to use this kind of approach to identify taxa that would be worth targeting for future study. And so I've just talked about PKSs. There are lots of other upgrades. Our NAPTOS version 2 is we're just upgrading the workflow for the KSs. We're planning on upgrading the workflow for the condensation domains as well. There are other opportunities to further expand this kind of approach. And in particular, there's another group of PKSs called type 3 PKSs, also known as chalcone synthases. These used to just be thought about as being in plants where they were well known. We now know they're widely distributed in bacteria, and a KS phylogeny for these KSs is also pretty informative. We can see lots of different clades corresponding to bacteria. We can delineate among these clades information about the size of the starter unit, which is important, and we can also distinguish plant from other eukaryote and prokaryote. So we think there's lots of opportunities to expand this to type 3s. This can certainly be expanded to PUFAs. Some work out of Eric Allen's lab 10 years ago looked at the KS phylogeny of environmental PUFA KSs and was able to identify, based on the clading patterns, those involved with making things like EPA and DHA, and lots of clades that we don't yet know what kinds of PUFAs they're making. So if you're interested in PUFAs, this would certainly be a great way to explore new diversity. We also think this can be extended to prenyltransferases as well. These are enzymes that add 5-carbon prenyl groups to natural products. This often enhances their biological activity. It's a really important aspect of natural product biosynthesis where there isn't really a good bioinformatic way to search for strains or environments that have the greatest potential to produce these prenylated molecules. We're particularly interested in new soluble ones that prenylate aromatic substrates. And so there are at least 3 clades here we can readily identify: bacterial prenyltransferases that prenylate phenol or phenocene, and fungal and bacterial prenyltransferases that prenylate a tryptophan-like molecule. And so there's other things we can also identify. It looks like prenyltransferases that act on ribosomal peptides and others as well. So really lots of great opportunities to expand this approach to make a very, I would say, facile way to screen large data sets to try to hone in on the types of samples or organisms that you think have the greatest potential to produce the kinds of molecules that you're interested in discovering. And I'll finally just say this also applies to halogenation. That's a really important part of bioactivity among natural products. And we've been looking at the flavin-dependent halogenases and looking at their phylogenetic relationships, which are predictive of the substrates that are prenylated. And so again, another potential application. So this is all really future thinking. Right now, we're just trying to get NAPTOS 2 out, which again is telling us, which upgrades the ketosynthase workflow. Again, this is a phylogeny-based classification scheme. If you detect a KS sequence and it looks a little different from anything in the database, we encourage you to put it into phylogenetic context. You may have a new clade, which may represent a new function. And the more functionality that's identified, the more we can enrich this type of approach. Again, applications, amplicons, genomes, metagenomes, doesn't matter what the source organism is or the environment. This is kind of a one-stop shop for this type of of biosynthetic prediction. And you can use this to help prioritize strains or environments. It might tell you where you want to culture from. It might tell you which strains you want to work with and try to induce expression of natural products. It also helps with dereplication. If you have a very good match to something in the databases, our experience has been there's a really good chance that your strain is going to produce compounds in that class. And so that could save you a lot of time and a lot of effort in doing all the chemical analysis workup. And so we're hoping to have this released by the end of the summer. That's our plan. And we look forward to people using it and giving us feedback on how to further improve it. And so finally, I've acknowledged a number of people along the way, in particular, Caitlin Creamer here, Lisa Clow here, who have been instrumental in developing this version 2 release, along with Sheila Padel, the programmer who's really been helping us do it. And my long colleague, Brad Moore, who I've worked with for many years on genome mining and his efforts in this regard as well. Okay, so I want to thank everyone for hanging in there if you were able to stick this out, and I hope you enjoy hearing a little bit about this web tool, and we'll find some applications for it in your research. So thank you very much for your attention.